From now we will see that the our program can be used to visualize and analyze the data generated by the chip manufactured by different companies like Affymetrix, Agilent and Illumina. So before going into the details how we can import the data to analyze the data generated by these chipsets, we have to know how these data is generated. So very briefly I will tell in few lines that if we want to see the genomics, we want to study the genomics data or genomic studies, we have to perform the whole genome studies, means the expression of all the genes present in the particular organism. For this purpose, we have to isolate the RNA first, then convert it into the cDNA if it is the eukaryote. If it is the prokaryotes, then we can proceed directly by isolating the DNA because there is no introns present within the prokaryotes. So if generally we use the expression data for humans, that is very important. So first we isolate the RNA, total RNA, and then we convert into the cDNA because we know that if the RNA number is present within the cell, for example, gene 1 has 500 RNAs, for example, gene 2 has the 30 RNAs, then when we convert the RNA to cDNA, the number will remain the same. Means the gene 1 has 500 RNAs, the number will remain the same when we will convert it into the cDNA. So now the cDNA has 500 molecules. So after converting into the cDNA, we compare, because we have to compare with the normal individuals, and these type of data, they are generally produced for some diseases, or if we are not using humans, then we can use the organisms which are treated with some chemicals or with other parameters. So it means we have to isolate these RNAs, then convert to cDNAs from the diseased humans, and then we have to compare it with the normal individuals. Then we will get the picture which genes they are upregulated or downregulated as compared to the normal humans. So after converting into the cDNA, we can label these cDNAs, both treated, the diseased and the normal, with two types of dyes. They are generally known as cyanin 3 and cyanin 5. One has the green color and other has the red color. So the cDNA of the normal is treated with the cyanin 3, that is green color, and the cDNA of the treated one or diseased one is treated with cyanin 5. So after treatment with these dyes, the color of the cDNAs will become either green for normal and red for disease. Then afterwards, we can just mix both of the samples from diseased and normal, because now we can, after mixing, can separate the number of the diseased and normal by the color, because we have two different colors and cDNAs have become either green or red. So after combining these two types of cDNAs, they are then hybridized with the probes. Probes, they are the small oligonucleotides, and each gene has at least one probe. So it means that if humans have 40,000 genes, then we have 40,000 probes that are already attached to a solid support, generally the small glass slide. So there is a small spot that is present for representing one probe, and they are present in triplicate. And then again the gene 2 has a probe, and it is present triplicate on the glass slide. So in short, they are hybridized with the sample of cDNA, and they will bind complementary with the sample of cDNA. So every gene has a different probes, and specific probes will bind with specific type of cDNA. And these cDNAs then visualize in the specific scanners, and then the expression of the data is analyzed by using the color of the cyanide dyes. So in short, I have told you, but now we will see the data that is generated by these type of chipsets, how they are imported. For example, if we use the Affymetrix data, so in the R package, the package Affy is present, and we can use this package to analyze the Affymetrix data. And we can use the command read Affymetrix, read Affy, to analyze the files that were generated with the Affymetrix chipsets. And these files are generated after, I told you, scanning of the hybridized chips. So the data that is generated, imported, it is saved in the form of the Affy batch object. And when we use these Affy batch objects, and if we apply the read Affy data, then all of the CEL, because we use the Affymetrix, the CEL files, they are generated. So all these CEL files, they will be read if we apply this read Affy command. And if we want to apply this command for a specific CEL files, then we have to specify which files we want to apply this read Affy command. So very briefly, we can see that the library will be generated. It is already present. Then when we use the read Affy command, data set is generated. Affy batch, it is saved in the Affy batch object form. And this we can see size of the arrays is 712 into 712. Feature is, this is the name of the array chip sets, and total of 22,283 Affy IDs. They are present in the form of probes. So the number of sample is 17, number of genes, they are 22,283, and the annotation name is this human GU133A. So we can see here that the data stored in this data Affy batch object is from human arrays, and it has 22,283 transcripts, 17 types of arrays, and some other information.